1.4 Enzymes. An enzyme is a biological protein-based catalyst. Enzyme nomenclature. Enzymes are divided into 6 groups based on the chemical reactions they catalyze. Oxidoreductases, transferases, hydrolyses, lyses, isomerases, ligases, synthetases. Enzymes online tutorial. Excellent animations. http://www.leoport.nirec.org/bannermaker/animations.htm http://www.northland.cc.mn.us/biology/11/animations/enzyme.html http://hide.mcgraw-hill.com/site/00724373116/student_view0/chapter8/animations.html Substrate, the reactant that an enzyme acts on when it catalyzes a chemical reaction. The induced model of enzyme-substrate interaction describes a protein as a dynamic molecule that changes its shape to better accommodate the substrate. Factors affecting enzyme activity: temperature, pH, substrate concentration. Enzyme concentration. 37 degrees Celsius is the optimum temperature in most living systems. At high/temperatures, enzyme function is altered. Denatured enzyme. Most enzymes work best at pH 7, narrow range. However, some such as pepsin have a pH of 3. Changing the pH will alter the enzyme's 3D shape. pH. Substrate concentration. As concentration increases, the rate of reaction increases. Maximum occurs when all the enzymes are working. Cofactors. Inorganic atoms, Zn, Ca, Fe, which bind temporarily to enzyme. Coenzyme organic molecules, for example, vitamins. Necessary to activate enzyme and allow it to bind to substrate. Also can help weaken bonds in substrates. Controlling enzyme activity. 1. Regulating transcription/translation. By regulating transcription/translation, production of enzymes can be turned off/on. Some end products can act as transcription factors and inhibit the transcription/translation of enzymes. 2. Competitive inhibition. A substrate mimic molecule that has the same configuration as the substrate can enter into active site and block enzyme action. Some antibiotics are competitive inhibitors. For example, HIV, protease inhibitors. Penicillin inhibits a bacteria transpeptidase. Cell wall is not built properly. When bind permanently, poisons or toxins. When bind temporarily, can be dislodged by increasing concentration of substrate. Inhibitors. Three. Allosteric inhibition, feedback inhibition. 3. Allosteric inhibition, feedback inhibition, allows an enzyme to be temporarily inactivated. Binding of an allosteric inhibitor changes the shape of the enzyme, inactivating it while the inhibitor is still bound. This mechanism is commonly employed in feedback inhibition. Often one of the products of a series of reactions act as an allosteric inhibitor and blocks the pathway, aka non-competitive inhibition. 3. Regulation of enzyme action. 4. Allosteric activators. Substances bind to an allosteric site on enzyme and increase efficiency. A. Substrate and enzyme. B. Competitive inhibitor. C. Non-competitive inhibitor. Enzymes are _catalysts, and as such they underscore a chemical reaction without being underscore in the process. Enzymes work by reducing the underscore. The underscore is a reactant that an enzyme acts on. This reactant binds to a particular spot on the enzyme known as the underscore. Enzymes are very underscore for the reactant to which they bind. The names of enzymes usually end in underscore. ANS, protein, speed up, consumed, activation energy, substrate, active site, specific, ACE. Temperature and pH affect enzyme activity. Temperature and pH affect enzyme activity. As with all other reactions, enzyme-catalyzed reactions underscore in speed with an increase in temperature. However, as the temperature increases beyond a critical point, the protein structure begins to get disrupted, resulting in underscore and loss of enzyme function. Every enzyme has an underscore temperature at which it works best, and activity tends to decrease on either side of this temperature. Most human enzymes work best at around underscore. Some enzymes require non-protein underscore, such as zinc and manganese ions. Other enzymes may require organic underscore such as NAD+ and NAD+. A variety of substances inhibit enzyme activity. Underscore are so similar to the enzyme substrate that they are able to enter the enzyme's active site and block the normal substrate from binding. This process is reversible and can be overcome by increasing the concentration of the enzyme substrate. Another class of inhibitors does not affect an enzyme at its active site. They are called underscore, and their effect cannot be overcome by adding more substrate. ANS, increase, denaturation, optimal, 37 degrees Celsius. Cofactors, coenzymes, competitive inhibitors, non-competitive inhibitors. Practice. 1. Define catalyst. A chemical that speeds up the rate of reaction without being consumed in the reaction. 2. Draw a labeled free energy diagram to illustrate the effect of an enzyme on the activation energy of a hypothetical reaction. Assume it is an exergonic reaction. 3. What is meant by the statement, an enzyme cannot affect the free energy change of a reaction? 5. How does an enzyme lower the activation energy of a biochemical reaction? The enzymes do this by bringing the substrates into the correct geometry and by putting stress on the necessary chemical bonds. 6. How do competitive enzyme inhibition and non-competitive enzyme inhibition differ? A competitive inhibitor binds to the active site of an enzyme, preventing the substrates from binding. In this case, the inhibitor competes with the substrate for the active site. A non-competitive inhibitor attaches to an enzyme at binding site other than the active site. This causes a conformational change in the enzyme's protein structure that causes a loss of affinity of the active site for its substrate. 8. What happens to an enzyme after it has catalyzed a reaction? After an enzyme catalyzes a reaction, it will catalyze the same reaction again.